All organisms, including plants and animals that we observe, are composed of cells. Each and every cell arises from its pre-existing cell and becomes specialized for performing different functions after division. Cell is the fundamental structural and functional unit of living organisms. Do you know who discovered cell and when? Robert Hooke discovered cell in the year 1665 while he was examining a thin slice of cork under his primitive microscope. He observed that cork consists of small box-like structures resembling honeycomb. He called these boxes cells. Cork comes from the bark of the tree. Cell is a Latin word for a little room. Before getting into any detail, we should first understand the basic characteristics of cell. They have the ability to replicate independently. They contain heredity information. They perform life-sustaining activities and they show similar chemical composition and metabolic activities. Let us see some major discoveries related to cell. Robert Hooke was a scientist who discovered cell in 1665 for the first time while he was examining a thin slice of cork with the help of his primitive microscope. Leeuwenhoek in 1674 discovered free living cells in pond water with his improved microscope. Robert Brown in the year 1831 discovered nucleus in the cell. Sheldon and Schwann in the year 1838 to 39 presented the cell theory, which states that all plants and animals are composed of cells and cell is the basic unit of life. Purkinje in the year 1839 coined the term protoplasm for the fluid substance of cell. Virchow in the year 1855 expanded the cell theory by suggesting that all cells arise from pre-existing cells. Knoll and Ruska in the year 1940 built an electron microscope that made understanding of complex structure of cell and its organelles possible. On the basis of number of cells present in organisms are classified into two types. First one is unicellular organisms and the next one is multicellular organisms. Unicellular organisms have single cell that constitutes the whole organism. Multiple cells are grouped in a single cell body that perform different functions. Whereas in multicellular organisms, there is no division of labor in prokaryotes, but in eukaryotes, division of labor is seen. What is division of labor? It means when different functions are performed by different parts or compartments. That is known as division of labor. And all cells are specialized to perform different functions of the body so that there is division of labor within a single cell as well as group of cells. Cells are of different shapes depending upon the function they have to perform. Some cells have fixed shapes, example plant and animal cells, while some cells like WBCs and amoeba keep changing their shapes. Fixed shapes can be of various types. Elliptical as in fat cells, spherical is ovum, spindle-shaped is found in smooth muscle cell, knobbed thread-like shape is found in sperm cells, discoidal is RBC, elongated cells are found in the nerve cells. The size of a single cell is also different. Longest cell is the human nerve cell that is of 90 cm. The smallest cell is of the mycoplasma. It is a bacteria. It is of 0.1 to 0.5 microns. And the largest cell is that of an ostrich's egg that is about 18 cm. Functions of cell. The shape and size of cells are related to the specific function they perform. Multicellular organisms perform these functions by division of labor, that is, different parts of human body are specialized to perform different functions such as heart is made to pump blood, stomach to digest food, and kidney to filter urine, etc. A division of labor is also seen within a single cell. Every cell possesses certain specific parts called cell organelles which enable it to live and perform special functions. Cell organelles together constitute the basic unit of life called the cell. Each kind of cell organelle performs a specific function like obtaining nutrition, respiration, clearing waste material, or forming new progeny. Example, mitochondria is the organelle responsible for providing energy to the cell. Structural organization of cell. Every cell possesses three basic features in common, viz. Plasma membrane, nucleus, and cytoplasm. Due to the presence of these features, all activities inside the cell with its environment are possible. Plasma membrane or cell membrane. This is the outermost covering of cell. It is thin and delicate. It separates the contents of the cell from its external environment. It is made up of lipids and proteins. Phospholipids. Phospholipids provide flexibility to cell membrane and enables the cell to engulf food and other materials from external environment. This process is called endocytosis. Amoeba acquires food through this process with the help of finger-like projections called pseudopodia. Functions of cell membrane. It allows the entry and exit of some selective materials in and out of the cell. The cell membrane therefore acts as semi-permeable or selectively permeable membrane. It helps to maintain the shape of the cell. It acts as a mechanical barrier and protects the internal contents of the cell from leaking out. It provides protection against microbes and foreign substances. It gets modified to perform different functions. Example, microvilli in the intestine of human beings for absorption. Its semi-permeability enables the cell to maintain cellular homeostasis. Homeostasis means to maintain a stable and constant environment inside the cell. Transportation of substances across the plasma membrane. Transportation of substances across the plasma membrane may take place with the expenditure of energy, that is active transport, or without the expenditure of energy, that is passive transport. Transportation across the membrane by diffusion. The spontaneous movement of a substance from a region of its higher concentration to a region of its lower concentration is called diffusion. For example, carbon dioxide accumulates in higher concentration inside the cell. In cell's external environment, the concentration of carbon dioxide is lower as compared to inside of the cell. Due to this difference in the concentration, carbon dioxide moves out of the cell by the process of diffusion. Similarly, oxygen enters the cell by the process of diffusion when the concentration of oxygen inside the cell decreases. Facilitated diffusion means passive diffusion. It means without the expenditure of energy, it takes place with certain proteins that are known as carrier proteins. Transportation of substances across the membrane by osmosis. The movement of water molecules through a selectively permeable membrane along the concentration gradient is called osmosis. Concentration gradient means concentration difference. Osmosis is defined as the passage of water from a region of high concentration to a region of low water concentration through a semi-permeable membrane. Osmosis can be seen in a cell placed in solution of different concentrations such as hypotonic, isotonic, and hypertonic. Hypotonic solution. When the surrounding solution has a higher concentration as compared to inside of the cell. As a result, the cell will gain water and swell up via endosmosis. Because there will be movement of water molecules from outside of the cell to the inside of the cell. Isotonic solution. When the surrounding medium has same concentration of water as that present inside the cell. Water crosses the membrane in both directions, but the amount moving in is the same as the amount moving out. So there is no overall change observed and the cell will stay in same size. Hypertonic solution. When the surrounding medium has a lower concentration of water than the cell, that is, outside solution is very concentrated. Water crosses the cell membrane in both directions, but this time more water leaves the cell than enters it. As a result, the cell will shrink. Cell wall. It is a tough non-living covering outside the plasma membrane. It is found only in plant cells. It is freely permeable. It is made up of cellulose that gives structural strength to plants. Functions of cell wall. Cell wall permits the cells of plants, fungi, and bacteria to withstand hypotonic conditions without bursting. It has narrow pores called pits through which fine strands of cytoplasm called plasmodesmata are able to cross the cell walls. Plant cells interact with each other through these cytoplasmic channels. Nucleus. It is called brain of the cell. It is composed of a double layered covering called nuclear membrane. It has numerous pores called nucleopores for the transfer of materials from inside the nucleus to cytoplasm. It contains a liquid ground substance called nucleoplasm which contains nucleolus and chromatin material. Nucleolus is a round structure found inside the nucleus and does not have covering membranes. It is called factory of ribosomes. Chromatin is an entangled network of long thread-like structures which condenses to form chromosomes during cell division. Functions of chromosomes. They contain information for the inheritance of features from parents to next generation in the form of DNA molecules. Chromosomes are composed of two components, that is DNA and protein. The functional segments of DNA are called genes. Functions of nucleus. It plays an important role in cellular reproduction in which cell divides to form two new cells. It determines cell development and maturity. It helps in the transmission of hereditary traits from parents to offspring. It controls all metabolic activities of cell, and if it is removed, the protoplasm dries up. There are two types of cells. One is prokaryotic and another is eukaryotic. In prokaryotic cells, pro means primitive, it means not advanced, and karyon means nucleus. Organisms whose cells lack a nuclear membrane are called prokaryotes. Prokaryotes also lack cytoplasmic organelles. Most functions are performed by poorly developed parts of the cytoplasm. In eukaryotic cells, U means true and karyon means nucleus. Organisms with cells having a well-defined nucleus enclosed in a nuclear membrane are called eukaryotes. Eukaryotic cells are of two types, plant cells and animal cells. There are many differences between a prokaryotic cell and a eukaryotic cell. The first one is the size. In prokaryotic cell, the size is generally small, 1 to 10 micrometers, and in eukaryotic cell, the range is of 5 to 100 micrometers. The nuclear region is poorly developed, no nuclear membrane is there, and it is called as a nucleoid. Whereas in a eukaryotic cell, well-defined nucleus surrounded by nuclear membrane is present. Single chromosome is there in prokaryotic cell, whereas in eukaryotic cells more than one chromosomes are there. Nucleolus is absent in prokaryotes, whereas in eukaryotes, nucleolus is present. Membrane-bound organelles are absent in prokaryotes, whereas in eukaryotes they are present. Centriole is absent in prokaryotes, whereas it is present in animal cells. The organisms in prokaryotes found in bacteria, blue-green algae, etc., whereas the organisms found in fungi, plants, and animals possess eukaryotic cell. The cell division in prokaryotes takes place by binary fission or budding, whereas in eukaryotes it takes place by mitotic or meiotic cell divisions. Eukaryotic cells are of two types, one is plant cell and another is animal cell. There are some differences between a plant cell and an animal cell. In plant cells, cell wall is present. Cytoplasm is not dense and a large vacuole is present. Plastids are present and centrosome is absent. Whereas if you look at animal cell, the cell wall is always absent. Cytoplasm is dense. Vacuoles are always absent. Plastids are absent and centrosome is always present. Cytoplasm. The large region of each cell enclosed by the cell membrane is called cytoplasm. It is the fluid content present inside the plasma membrane. Many cell organelles are embedded in it that perform specific functions. Functions of cytoplasm. It helps in the exchange of materials between organelles. It acts as a storehouse of vital molecules such as amino acids, glucose, vitamin, iron, etc. It acts as the site for certain metabolic pathways such as glycolysis, etc. What are cell organelles? The membrane-bound structures that perform specific functions are called cell organelles. This is the main characteristic feature that separates eukaryotic and prokaryotic cells. The first cell organelle that we are going to study is the endoplasmic reticulum. It is a large network of membrane-bound tubes and sheaths extending from outer nuclear membrane into cytoplasm. It occurs in three forms: cisternae, vesicles, and tubules. Depending on nature of its membrane, ER is of two types: rough ER and smooth ER. Rough endoplasmic reticulum RER. It contains ribosomal particles due to which its surface is rough. The ribosomes are the sites of protein synthesis. RER is mainly formed of cisternae. Its surface is smooth due to absence of ribosomes. Smooth endoplasmic reticulum SER. It helps in manufacture of fat molecules or lipids. It is formed of vesicles and tubules. Its surface is smooth due to absence of ribosomes. Functions of endoplasmic reticulum. Ribosomes present in all active cells act as sites for protein synthesis. Proteins manufactured here are transported throughout the cell by ER. Fat and lipid molecules manufactured by SER helps in building cell membrane. This process is called membrane biogenesis. Some other proteins and lipids synthesized by ER function as enzymes and hormones. SER plays a crucial role in detoxification of poisons and drugs in liver cells of vertebrates. It forms a network system providing channels for the transport of materials, especially proteins, between various regions of the cytoplasm and the nucleus. It also functions as cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. Golgi apparatus. It consists of a system of membrane-bound, fluid-filled vesicles, large spherical vacuoles, and smooth flattened cisternae which are stacked parallel to each other. Each of these stacks is called a cistern. Functions of Golgi apparatus. The material that is synthesized near ER is packaged and dispatched to various parts, that is inside and outside the cell, through Golgi apparatus. It stores, modifies and packs products in vesicles. It is involved in the formation of lysosomes. It forms complex sugars from simple sugars. It is involved in the synthesis of cell wall and plasma membrane. Lysosomes. These are a kind of waste disposal system of the cell. These are membrane-bound sacs that are filled with digestive enzymes. These enzymes are made by RER. They are called suicide bags of cell because when the cell gets damaged, lysosomes may burst and the enzymes can digest their own cell. They are absent in RBCs. Functions of lysosomes. They help to keep the cell clean by digesting any foreign material that enters the cell as well as worn out cellular organelles. Hence, they are called scavengers and cellular housekeepers. They remove bacteria or any foreign material entering the cell by breaking it into small pieces through its powerful digestive enzymes, which can break down all organic materials. During starvation, lysosomes digest stored food contents by autophagy and supply energy to the cell. Mitochondria. It is a double membrane-bound structure. The outer membrane is very porous while the inner membrane is deeply folded into finger-like projections called cristae, which create large surface area for ATP generating chemical reactions. Space between outer and inner membrane is called intermembranous space. It is self-replicating organelle. It has its own DNA. It is the largest organelle in animal cells. Mitochondria is a very important topic and one question is definitely asked in the exam from this topic. So make sure that you do it thoroughly. Functions of mitochondria. It generates energy for various activities of cell. Mitochondria are sites of cellular respiration and release the energy required by the cell in the form of ATP adenosine triphosphate molecules. This ATP is called energy currency of cell. Whenever cell requires energy, ATP breaks down to release energy. Mitochondria are strange organelles in the sense that they have their own DNA and ribosomes, hence are able to make some of their own proteins. They help in the synthesis of various chemicals like fatty acids, steroids, amino acids, etc. Plastids. They are found only in plant cells. There are numerous membrane layers embedded in a material called stroma. Plastids are similar to mitochondria in external structure. They are also double layered and contain their own DNA and ribosomes. There are three types of plastids. First one is chloroplast, second is chromoplast, and third one is leucoplasts. Chloroplasts. These are plastids containing chlorophyll which gives green color to the plant. Chloroplasts are called kitchen of the cells. Function. These are important for photosynthesis in plants. Leucoplasts. These are white or colorless plastids. They can change into other type of plastids. Function. They store materials such as starch, oils and protein granules. Chromoplasts. These are colored plastids except green. Function. They impart color to flowers and fruits. They are rich in carotenoid pigments and lipids. Vacuoles. These are the storage bags for solid or liquid contents. In animal cells, vacuoles are small sized, but in plants, vacuoles are large sized and some may occupy 50 to 90% of the total cell volume. Vacuoles are bounded by a membrane called tonoplast. Functions of vacuoles. Vacuoles are full of cell sap and provide rigidity and turgidity to the cell in plants. Many substances like amino acids, sugars, organic acids and proteins are stored in vacuoles. In amoeba, consumed food items are stored in food vacuoles. In some unicellular organisms, vacuoles also play an important role in expelling excess water and some wastes from the cell.